You're listening to NASA in Silicon Valley, episode 70, and for the intro, I have Abby here with me again.
Hello, hello.
And this is a slightly interesting episode, just for the sake that when we planned on recording this, I got horrifically ill, and Abby jumped in at the last minute to go ahead and do the recording.
That's right.
So Abby, tell us about the conversation that you had.
All right, well, it turned out to be very interesting. I met with Sylvain Coste. He is the manager of the Gene Lab project here at Ames.
Okay.
So you know how NASA does a lot of biosciences. We do biology experiments up on the space station. So when those experiments end, the science doesn't end because all that data goes into a repository, which is open to the public. It's open access for any researchers to use. And right now they're developing tools and really building a system around it where people can come analyze this space biology data that NASA helped produce and looking for discoveries that they can make within it themselves. And this is for researchers, if that's your research, or for citizen scientists who may be interested, they'll be able to explore as well and get more out of the data than ever. So what Sylvain describes it as is NASA as the custodian of knowledge about how life is affected in space, which I think is cool.
Sounds super exciting.
Yeah.
So before we jump on into it, a reminder for folks listening, we have a phone number that's 650-604-1400. Give us a call and leave a message and we'll try to add that into future episodes. If you want to be digital, we are on all the social media platforms. We're using the hashtag NASA Silicon Valley. We are a NASA podcast, but we are not the only NASA podcast. So give a quick little shout outs to some of our friends over at headquarters who do Gravity Assist. There's also another weekly podcast called This Week at NASA. And then, of course, our friends over at JSC, over at the Johnson Space Center. They have Houston, we have a podcast. So that's where you can find all of our stuff. But for today,
let's listen to Sylvain Coste. So, hey, Sylvain, thanks for coming in.
Well, thank you for having me.
I'm excited to hear about your work a little bit. So usually we start this off by learning about you and your background and how did you end up at NASA? Where do you come from originally?
Sure. So, I was born in France and I went through physics and mathematics training in France in the college. And
cool.
Eventually I transferred to, I did an exchange at Texas A&M University. And from there I kind of liked the American education.
Yeah.
So I decided to go for a PhD. So after a master at Texas A&M, I did a PhD at UC Berkeley and spent some time at NCI, National Cancer Institute, and then became an independent investigator at Lawrence Berkeley National Lab, which is a DOE lab. And I joined NASA only last December in 2016.
Oh, so you're pretty new. You're even newer than I am to NASA. Interesting.
Good.
And I lived in Paris for 8 years, so we have something else in common.
So, je vais parler français alors. Hein?
Ah, non, peut-être pas aujourd'hui. Let's continue in English. So, from France, what part of France?
It's hard to tell. I mean, I was born in Bourges, which is one of the center city. But I moved, I think, 20 times. By the age of 20, I had moved 20 times in France. So,
oh my gosh.
I don't have any really city. I would say I'm from the south. That's really where my family is from. So Toulouse would be.
I see.
And the real city is called Rodez. But I don't really feel
Toulouse.
Like I'm just French in general.
Right. Okay.
I've been everywhere in France.
So you're used to moving around.
That's right.
Like. So from France to Texas to California, you've been all over.
Yeah.
And eventually that led you to NASA. But so you're a biologist, is that right?
No, I'm a physicist.
A physicist.
So my PhD is in nuclear engineering.
Oh my gosh.
I used to do, in my master, I was doing nuclear reactor design, so a lot of neutronics. And then in nuclear engineering, there is a section called health physics, which is understanding how radiation impacts people's life. So like how you can get cancer from radiation, the risk of ionizing radiation. I got into this. And part of that is called medical physics, which is understanding how radiation can be used to treat cancer.
Mm-hmm. That's the connection between physics and cancer research.
That's right.
Yeah.
And then little by little, I kind of left the physics world to move more and more towards the biology. So being a physicist, I've done a lot of I've used a lot of the physics knowledge, so mathematics and modeling, into understanding biological processes. Until last year, very much focused on radiation. And so the connection with NASA there was that there is cosmic radiation that astronauts get exposed to. So I've been studying their impact on humans for a long time.
Right. Radiation's a big challenge.
It's
for
a big
space
one.
Exploration.
Microgravity and radiation are the two big ones, right?
Right.
And now at GeneLab, I'm really emphasizing everything. So radiation is just one small aspect of what we're working on. We're really looking at the full response of the human on, life in general, in terms of living in space.
Right.
So microgravity.
The physical effects of
absolutely.
the space environment, right?
That's right.
Right. Okay. So you just mentioned GeneLab. What is that?
So GeneLab I'm the project manager now for GeneLab. And so let me tell you a bit about GeneLab, because it's a project that started about 4 years ago, roughly. And the idea, which is, I think, very good, is that NASA should be the custodian of the knowledge of how life gets impacted in space. And so there's a lot of studies that have been going on for 20, 30 years under the sponsorship of NASA. And
definitely, we've got a big biosciences division here. Right?
Absolutely. And so there's a lot of scattered information here and there. And I think we're lucky to live in a time where now we have this new technology called omics. And so the omics are, you hear about them every day. It's actually, they're looking at specific changes in your genome called variants, and they're trying to interpret the slight different changes in your gene in your gene sequencing with respect to some risk to your health. That's one omics that's exploding right now in the world. But there's other omics that have been going on for a while. One of them is called transcriptomic, which is 90% of the data in GeneLab are transcriptomic data, and there it's the idea of looking at the RNA expression in tissues or in microbes or anything. We're looking at any sample coming from the space station or from the space shuttle end up, if they have been analyzed for omics, they end up into our repository. So GeneLab is that big repository of information.
Okay, right. GeneLab is a database.
It's a database, but it's going to be more than a database. This was the original thought for it. Basically, let's store all this information into one local place. And so we've been very active in either identifying legacy data set that should be in GeneLab from the get-go, from the past. And we're also very active with any new omics being produced on the ISS to make sure that those data comes into our repository. And we're not only looking at one type of omics, we're looking at many different omics. There's something else called proteomics, which is protein profile. Epigenetic, which is how your DNA gets decorated by specific molecules that changes the expression profile of those molecules. So all these omics techniques are coming to us, and we're working very actively also in identifying what's been already produced in the world. And the idea is to be kind of like the custodian of knowledge and catering this information to the public.
Okay, cool. So let's review. Omics is this big area, right? That it can be proteomics, genomics.
Absolutely.
Right. Okay. So any of these companies that are offering genetic analyses for
it's one type of
public,
omics.
Yeah, that's one type of omics. So all of these basically are ways to study what our DNA is producing or what any organism's cells are doing
right.
with their DNA. And is it correct that that's what can be influenced by the space environment?
Absolutely. So I think if you go back about life in space, there's really two big questions that we need to address. One is really how microgravity confinement, ionizing radiation can affect living entities. And here, that question is important with respect to the astronauts because we want to make sure they're going to be healthy on the long run.
Yeah.
So that's one big question. And we believe that in addition to the battery of tests you can do on an astronaut, like, blood samples, pulse, EKG, whatever, you have other tools, molecular tools that we can address by using animal models. So because the problem with omics is typically you have to sacrifice the animal to get the information.
I see.
So for a mouse, we're going to get the liver, we're going to get the brain, we're going to get the bones, and we can then run omics on those guys.
Okay. So to see what impact
When
the
it- so-
microgravity or radiation is having on the body.
on a body, right? That is
Yeah.
close to us, which is mammals
Yeah.
are great, but there's also effort on Drosophila, which is insect, and
The fruit flies.
fruit flies, and you also have C. elegans. So there's a variety of animal models you can use. There is another question that GeneLab is also helping answering in terms of the information we're putting into the database. It's more understanding the way an ecosystem is modified by space. And so here you can imagine microbes. So, we talk about microbes being found on the walls of the space station.
Yeah, I've seen that.
These kind of things would be addressable with omics, but more then you're more interested in seeing what kind of maybe new species or how a strain can deviate from its original genomic makeup by being in space for a long period of time. It's also helpful for the plant, for instance, understanding the kind of stress you put on an ecosystem like a plant.
Yeah.
Then you may be able to optimize how a plant grows on Mars or in the space station. So you see, you can either look at the ecosystem side or you can look at the human health side.
Yeah.
And so those two things are coming together in GeneLab, which is interesting.
That's very interesting. That's what I just realized as you were talking. You can look at the effect of space on an individual and then all the way up to the ecosystem scale.
Exactly.
That's pretty awesome.
Right.
Okay, so GeneLab, these studies are going on already, and then GeneLab collects all that data that these experiments are producing. Is that it?
Yeah, so there's different ways the data can make their way in GeneLab. So one thing, as I mentioned earlier, was we talk about legacy data. So before GeneLab existed, people were already gathering some omics. Having said that, the omics have changed a lot over the past 10 years. So the legacy data typically have some kind of technology for transcriptomics that we don't use as much anymore, like we call microarray, which is the old way of looking at RNA levels. Since then, gene expression levels. So since then, now we have RNA sequencing, which is a better technique. As we go to new omics, we have much larger data sets. So the repository is getting bigger, so big data is coming down the pipe.
Yeah, totally.
And that's one way, but the other aspect of GeneLab is to really work actively with investigators and collaborators to generate new data. And so we work with PI that have their funding from NASA to fly animal models, plant, microbes in the space station. And we help them maybe get more information from their samples and make sure that all their omics go on the database at the end.
This is the side that I know a little bit about from working at Ames, our bioscience department. They work with researchers at other institutions, right?
Right.
Who want to fly an experiment to space to do their science. That's what you're talking about,
Absolutely.
right? We'll help them carry out that experiment, and then also we get to use the data.
Absolutely. So there is that aspect of at NASA Ames where investigator will work with a principal investigator from that got funding to actually fly on the ISS specific mission and specific experiment. So part of our work is also there. And I think the something new about GeneLab that as the new project manager I'm trying to push for is really, I really think that GeneLab should be serving 3 different communities. So the data repository by itself, really, that data really talks to the specialist, the bioinformatician that can go in there and download the data and work with the data and interpret the data.
Specialists, yeah.
It's very, very specialized people, scientists. But then you have another group, which is the scientists in general, which they don't know how to do the bioinformatics, but they know how to ask the right question. And we want to provide tools for them to be able to access the information without having to do all these very tedious and slow work. So some of the repository data are now being used to be processed to generate a new level of data that we would call higher order data that can be interpreted. And from there, for instance, the idea would be, is there a signature of cancer in some liver samples that I got from the space station? So a specialist on cancer, but not a specialist in bioinformatics, can ask this question by being provided the right information.
I see. And they're also, that means they're not doing a brand new experiment. They're using data that exists.
Exactly.
Cool.
And so now you can think of, this, it's the same data, but they've already been pre-processed by us. And then they are now, so there's a bigger emphasis on tools to visualize this information. And so we're still working with this and with the investigator. And the idea would be to really have at the end, even a higher level type of information that would be very succinct but very simple to access. With a few clicks, someone could go in there and ask for their favorite gene. Say, is P53 modified in space? And then you could ask to look at all the mouse data, or you could say,
yeah.
okay, I want mouse and Drosophila. Or
Yeah.
and this now talks to not only scientists, but really even high school students can do these kind of
Really?
questions. Yeah, we
Yes.
had a gene lab for high school. Liz Blabber was the PI who actually organized this, and it was very successful. And you know,
Awesome.
high school student can make sense out of this data with the
Wow.
right guidance. So it's
Wow,
possible.
That's impressive. That's
Yeah.
bioinformatics, you're saying. High school students are working on
Absolutely, yeah. It's really
Awesome.
amazing. The new generation is well trained.
that. Wow, yeah. NASA's getting the next generation ready. That's awesome.
Yeah, there were like 20 plus kids that came here this summer, and they spent 3 weeks, and
Wow.
they worked on the data that was on the database, and they actually at the end they did a presentation, and
Wow.
it was really good.
That's a good way to spend your summer as a high school student.
I agree.
Not just hanging out at the beach, right? So you said 3 communities that Gene Lab serves. Did we hit all 3?
Yeah, those visualization aspect. There's really you could have a visualization layer that would be fairly sophisticated for still scientist type of people, but then you could really have even a higher level of visualization that is really simple, where you can ask very simple question about anyone who doesn't know science but who's curious about space could say, is there any change in inflammatory response in space? And
Okay.
so at least on the omics level, looking at protein and RNA, you could extract this kind of information actually and report this information back to the public. So what we're envisioning is really this multi-tier level where you can really, for specialists, you would pretty much play with the data the way they are. For the scientist community, you would have visualization tool and some processing tool. So if you want to do some grinding yourself,
Okay. Crunch
and
the numbers
crunch
from
the
that
number.
data. Yeah.
and this system would be, by the way, on the cloud. But then at the end, the very light level data, which doesn't take much room, is those visualization data. But then they're very much guided by us, because we have to make choices in what we want to display. And
Right.
and so, and to do this, we think that we will involve the community, the scientific community, through something. This NIH has used a similar model called AWG, Analysis Working Group. So the idea is to put together multiple principal investigator experts in one topic, and put them together to kind of tease out what would be the best way to analyze certain type of data. So, for instance, we could focus on the rodent data, or you could focus on the microbe data. And so you could imagine different AWG, Analysis Working Group, for these different questions.
Okay.
And
So different groups of scientists will decide this particular kind of data is probably most useful for the community, so we're going to create some tools to process it.
some tools or some way of displaying them.
Okay.
So how can you make it very, very easy for anyone to understand what's going on? So what is the right processing pipeline? So we call them pipeline. It's like it's a bunch of different scripts that you put together that will take the raw data, which are very big, and turn them into a very small amount of data that is small but very meaningful to us. You can imagine Google does the same thing with their data. They have all these very large database that they work with, but at the end, when you type a keyword for a specific question you're asking, the system is able to point immediately to a web page with an actual answer to your question, which is remarkable. So in the background, there's a lot of things happening for this, and there's that huge database working for you.
Are you kind of creating the search engine that will browse this huge database of biological
We're thinking of
information?
that. It's a bit more difficult for us because when you think of Google, they really have what they call big data, which is real. It's a lot of data. Gene Lab doesn't have big data. We have complex data. We have,
okay.
but we don't have that many experiment from space mission. And if you go on our website right now, you will find 130 plus studies, and less than half of them are actually space mission, and the other one are ground studies that mimic what's happening in space.
Oh, okay. Simulation.
Space. So this is the caveat, is that we have lots of data, but they are complex data and they're not big data. We have a very sparse matrix of information. And so there's still some question about how you're going to go about those data. And so that's really where working with the scientific community will help us figure out what are the best pipeline with this specific constraint in mind, which is an additional challenge. But I think the technology and I think machine learning may be helpful there.
Really? Machine learning is part of this?
We're thinking of that too.
Interesting.
because there might be some clever way of interpreting those sparse metrics that we're dealing with. And there's a lot of things still that are, undefined in the scientific community. And I think GenLab is really at the cutting edge of this information. So it's super exciting, but it's a challenge. And I think it's a visionary approach to have created GenLab. But any visionary approach also bring a lot of challenges that needs to be dealt with.
Well, yeah. But NASA's all about challenges
Absolutely.
and taking them on.
That's what we hear, right?
Right, exactly. I wanted to ask you, you've spoken about how GenLab will be accessible to different levels of expertise, right? Is it also open to anyone to go browse and look at?
Absolutely. So the intent is to have this different tier level. Currently, the current version we have is 1.0, and we're moving to 2.0. 1.0 is very much a repository where you can just download the data. The version 2.0 would have, and it's public. So, anyone can go in there. There is no restriction.
Amazing.
So a high school student can download the data on his or her hard drive and play with them.
Cool.
If they want to. There's a lot of, free tools out there that you can do that, really. But 2.0 is going to have more interesting things coming down the pipe. We have now a workspace, so people can log in, can actually see all your data that you want to add to the current GenLab data. So you can bring your own data to compare. If you want to do a comparison, for instance, with your favorite experiment and some space samples, you can do it inside the system. And the other thing is 2.0 will be, as we move on, we'll be having more and more tools that you can use to process some samples. So and do some analysis.
Does that mean like a cancer researcher could take their own data from their own lab and compare genetic changes to what we see in space?
Absolutely. That's exactly the idea. So you could have someone who's a specialist in breast cancer. And we know, for instance, breast is a very sensitive tissue for radiation. So it's a classic model. So it would not be a bad idea to look at and a lot of the animals that were flown on the ISS and in the space shuttle are female mice. For many reasons, it's easier to work with female than male. Typically, male tend to fight in the same cage for a start. So we can't put as many males as we can put female in a cage.
Yeah.
And so the female have the mammary gland, which is another very interesting tissue because they're very sensitive to radiation. So you could look at cancer incidence through radiation. So it would be an interesting question to look at specific early onset of cancer signature at the genomic level, and then compare it to the space station data that's on GenLab, for instance. I don't think anyone has done that yet.
Interesting. All right, and GenLab would make that comparison possible.
Should be. If we have, having said that, we need to first have some mammary gland data in GenLab for ISS or Space Shuttle, and they might be out there somewhere. A lot of the data are being generated by the PI as we're talking, so there's more data that are going to come along, and hopefully some of this information will be there. The longer we wait, the more information there will be there.
Right. It's going to grow with time.
Exactly.
Yeah. Cool. So do you guys just receive data, or do you ever work with the samples that come back from the space station? Because there are biological experiments happening up there, right?
It's a great question. So actually we do both. I mean, the majority of the work is obviously on taking other people's data. But NASA has recognized that some samples may not be taken by any PI, and so it would be a bit of a waste.
They may not be used by...
Right. And so GenLab has come up with a prioritization of samples that we think are very important. One of the strategies would be that if we can really focus our attention to specific tissue on a regular basis, then we'll have a very clear characterization of this tissue. And as time goes by, we'll have multiple time points in space. So a long duration versus a short duration, looking always at the same tissue in the same type of animals, then we'll be able to see how the time dependence is showing up. So to do this, we have what we call the sample processing lab, which is a small group in GenLab that either works with other principal investigators when they need help to process samples, but also there's something called tissue sharing agreement where we can get some tissue from the ISS that are not claimed by anyone else. And there's a list of tissue that would rather see coming in through this prioritization.
What would be an example? What would be tissues you're interested in?
So the one we've been looking at a lot is liver. And the reason no one wanted to look at liver is because it's not a tissue that's been showing very much response. Having said that, we actually now have a publication being prepared on that topic showing that actually there is some real changes in the liver in space.
Oh, really? Ah.
Which is surprising. There was one study before that had suggested there was a change and a longer duration from space shuttle samples, and now the study we're preparing actually is showing that on the ISS as well, the same strain of mice called C57 are showing some kind of a change in the liver over a 30-day course in space. So
that could be important for human astronauts.
Absolutely,
later,
right? The problem with animal system is that you have to remember that we work with one strain, which means that all the data is coming from one single strain, which is the equivalent of when you work on one strain of mice, you're looking at identical twins, if you want.
Oh, yeah. Okay.
So you have no idea of how genetic variance is affecting this response. So what you see in one strain may not be seen in another strain. And so that's one of the big challenges with the animal work.
I see.
So that's a caveat. And that's why insects are pretty cool, because with insects, you can actually have a bunch of different genetic background in one experiment.
Oh, more easily, more of them. They're smaller. Easier.
That's right. So you see, this is the art of science. It's like, how do you use each model to their best? How do you optimize the usage of these animals?
Yeah,
so
put them to their best use. Yeah.
Right. So rodents are great because they're very close to us genetically, but that's the limitation. Insects are great because, like Drosophila, you can have a huge spectrum of genetic differences, and you can have many of them, but then they're much further away from us than a mammal.
Yeah,
so, and then we put all this information together. The idea, again, as we move forward with technology, we expect to see some new algorithm that'll be able to make these bridges between the different species and come up with some real response from space and understand better how space affects us.
Yeah, right. Okay, so take the results from those studies happening in space, look at the data in a broad way, and draw conclusions.
Absolutely.
Okay, that's super interesting.
It is, right?
Yeah, cool.
10 years from now, we can go back and see what we discover, but I think there's going to be a lot of discovery by the scientific community through this database.
Yeah, no doubt. And the other thing I love about the space station biology experiments is it's not just for space applications, but everything we learn about human health from that can be applied down here potentially.
Absolutely. That's a great point. Thanks for raising it, because we're discovering this as one of the things we're doing right now at GeneLab is as we are kind of generating those pre-processed files for opening the door to a bigger community that don't need to do all this processing that we can provide to them. We're discovering some confounding factors in the sample. So for instance, if you modify the carbon dioxide level in the cage of an animal, so I don't know if you know that, but carbon dioxide levels are different in space because it tends to be higher.
Oh, really?
And for the longest time, we thought that there was no impact because they were still pretty low level. And now with the GeneLab data, what we're discovering is that when you do a ground control and you increase the carbon dioxide to the level that you have in space and the space station, we do see some hypoxic signatures in the gene.
It has an impact then.
It has an impact. Now, again, you have to be careful. RNA level is just one very small piece of the puzzle. You may have a change at the RNA level, but not at the protein level, which is what's more relevant, I would say, physiologically, is like the final signal is turned into an actual protein. So there's caveats in everything we do, but it's really telling us that, those carbon dioxide have an impact. It's not maybe picked up, physiological changes are maybe not picked up by it.
Okay,
But those very sensitive molecular tools can pick up those features.
Right. So that's an example where your GeneLab is allowing you to discover that it's very complex, the interactions between environment and DNA and proteins produced.
Exactly.
And you're teasing that apart, right?
Right, because back to the carbon dioxide example, you can imagine a situation on Earth where we are exposed to high level of carbon dioxide and no one would ever study this stuff because no one would ever think of that. But it turns out that this data set is clearly putting their fingers on one thing that maybe suggesting more and more studies even on by other investigators. What are those signatures? What are those changes in the RNA will do on the long term? Is there a situation on Earth where you get low carbon dioxide level and you should be concerned about it? So it is really going much more beyond space. The people being bedridden for like months is the equivalent of being in microgravity. That's one of the classic model. Well, microgravity can tell us about bone loss and things like this. So
that's right.
There's a lot of parallel between what's happening in space. You can think of space as an accelerator of aging, in a way. That's the way I look at it often. And so I think, everything we're discovering on those data will be relevant for human on Earth as well.
Fascinating. And I like the way earlier you described NASA as the custodian of data about biology and physiology and health in space. And it sounds like you're making that easier to use and accessible to more people.
That's what we're trying to do.
Wonderful. Excellent. Well, this was super fascinating. And I think
Thank
for
you.
a lot of people, it's surprising, first of all, that NASA does biology, and then that they can take a look at this data and maybe use it themselves in their labs or at home. So thank you for sharing that with us.
Well, no, thank you for highlighting GeneLab, and anyone who's listening, feel free to come to GeneLab.nasa.gov.
Awesome. And also online, we are at NASA Ames, and we can take any questions for Sylvain about GeneLab with the hashtag NASA Silicon Valley. So thanks again for being here.
Thank you very much.